Fossils. Fossils are formed in a variety of ways, depending upon the organic materials involved and the environmental conditions. 1. Entire organism preserved. In a continuous dry or cold regions, all organic remains, even the softer body parts, may remain unchanged for a long time under exceptionally favorable conditions. Organisms may be preserved intact in a medium that protects them from decay by bacterial action. Among the most perfect fossils known are the insects preserved in amber, a fossil resin from pines, especially Picea succinifera. Millions of years ago, insects such as bugs, aphids, caddisfly, mayfly, etc., became entangled and entombed in soft, sticky resin exuding from pine trees. The resin hardened and eventually changed to amber, preserving the minutest details of structure of the contained insects. A few extinct animals are known from frozen specimens in which flesh and bones have been preserved in remarkable fresh condition for thousands of years. For example, woolly mammoth has occurred principally in Arctic tundra, Siberia. Likewise, intact fossils of elephas and mastodon have been obtained from oil-rich swampy soils from Los Angeles, California. A ground sloth was found well preserved in an extinct volcanic crater in New Mexico in 1928. The skeleton of this Pleistocene mammal is complete. 2. Original hard parts of invertebrates preserved. In case of invertebrate animals and angiospermic plants, the harder parts such as shells, chitinous exoskeleton or spicules of animals and woody tissue of plants get preserved and fossilized. 3. Skeletons preserved. The skeletons of vertebrates make perfect fossils because they could be well preserved in their original shapes and structure. 4. Altered hard parts of organism preserved. Sometimes the original hard parts of an organism get completely altered due to various factors. Carbonization. Occasionally the disinterfossil perch. Grating soft parts of a body leave behind a thin film of carbon. Due to carbonization, have been obtained exact body outlines of the extinct swimming reptiles such as Ichthyosaurus and of other organisms such as jellyfishes, worms, leaves, etc. Petrifaction. When the mineral-rich water penetrates through the pores left behind by decomposition of organic matter of the harder parts, then minerals get deposited in these pores. The process is called petrifaction or permineralization. The deposition of minerals makes these fossils of harder parts more resistant to weathering agents. Sometimes, the histological or finer structures such as fibers, wood earth pores and lamellae in shells are perfectly preserved. This is called histometabasis. Mineralization. Sometimes certain specific structures of the fossil remains are replaced by a specific mineral. This process is called mineralization or replacement, e.g., 1. Mineralization of valves of vessels by silica instead of collagen. 2. Mineralization of sieve tubes, vessels and cells of woody tissues of plants. Mineralization involves more than 50 minerals. Some common examples are the following. 1. Carbonate of lime or calcium carbonate (CaCO) replaces siliceous parts of sponge spicule. 2. Silicor silicon oxide (SiO) is deposited in rings around a central nucleus in hair or nails, etc. The rings are known as Biekite rings. 3. Iron oxide replaces the original material and converts it into hematite Fe(O) or limonite Fe(O) NH(0). 4. Tron sulfide replaces calcareous parts of mollusks, crustaceans and brachiopods and converts them into pyrites, iron disulfide, Fes, and marcasites. Iron silicate replaces the tests of foraminifera and shells of molluscans. These replaced fossils are called glauconites, silicates of iron. V carbonates of Ca and Mg alter the original skeleton of corals, brachiopods and echinoderms. Such fossils are called dolomites. 5. Traces of organisms preserved. Occasionally neither an entire organism nor a part of it is fossilized. Instead, the organism left certain impressions or marks in certain hard media. They too bear great paleontological significance and are of the following types. Molds and casts. A mold is an impression of some hard parts, i.e., shell of an organism buried in the sediment or mud, which, in course of time, harden to become a rock. Later on, the organism decays, the organic material is removed by percolating acidified water and a cavity, pit or depression, is formed that shows the external configurations and surface marking of the original material. It is called external mold. Sometimes, however, the internal cavity of part, e.g., shell, is filled with mud or sediment or mineral deposits before the shell is dissolved by the action of percolating water. In such cases these molds are called internal molds. These represent internal structure of the shell and the exact shape of the animal. The molds are available largely for molluscan shells, foraminifera and radiolaria. If the mold showing external form, later on, is filled with mineral matter, natural cast of the original object is produced which reflects only its external form. Natural molds and casts form a very large proportion of the fossils that are found in sandstones and limestones. For the preparation of artificial casts, latex rubber solution, melted wax or plaster of Paris is filled in the external mold. Natural molds of several men and dogs, whose artificial casts have been produced by plaster of Paris, have been found in the ancient ash-covered town of Pompeii where they were mass buried by the eruption of the Mt. Vesuvius in 79 AD. Tracks and trails. Tracks, footprints, are formed over dryland or sea bottom or in muddy environments, where wet sand and mud receive impressions of the feet of passing animal and they are covered by the sediment before they are disturbed or eroded. Such fossils of footprints are very common for dinosaur reptiles from Triassic rocks of Connecticut Valley. The tracks of Mississippian amphibians form another example of track. With the help of these tracks, paleontologists gather information regarding the size and shape of foot, length of limb and posture and gait of the animal concerned. Trails are the irregular markings of moving animals on the sedimentary rocks, such as those formed due to the crawling of a worm or snail, the dragging tentacles of a jellyfish, the impressions of the fins of fish, and markings of the movements of crustaceans or urchins. E. Burrows and borings. Some animals live in burrows, tubes and holes in the ground, wood or rocks either for taking shelter or in search of food. The presence of such burrows or tubes in the sedimentary rocks of ancient past are called fossil burrows. They belong to bivalve and other mollusks, worms and sponges. Fossil burrows provide us information regarding behavior of their resident animals. 2. Skin impression. I. Borings are holes made by animals for the (I3) bone's sake of food and shelter. Such fossil holes occur on fossil shells, wood and organic objects. 1. Eggs in nest. IV. Gastroliths and coprolites. Gastroliths are hard stony pieces found from stomachs of ancient reptiles and fishes. Coprolites are fossilized feces of ancient animals. Their study provides valuable information about the food and feeding habits of fossil forms to whom the feces belong. Microfossils. One should not get the IM4 fossilized feces impression from our discussion that fossils are all coprolites, large, for many specimens are microfossils. In 5 footprint E, some fossils, e.g., spores, pollen, are only observable by light or electron microscope. Various types of fossils of the dinosaurs. Chemical fossils. In recent years, biochemists have joined forces with geochemists and paleontologists to track down a new variety of fossils, called chemical fossils. These are organic molecules that have survived relatively unmodified since they were formed by ancient organisms. Pseudofossils. Many objects of inorganic origin closely resemble the forms of organic origin and are found in the sedimentary rocks. They are referred to as pseudofossils. Significance of fossils. The study of fossils has great significance. It helps in understanding the prehistoric forms, process of organic evolution, and in reconstructing paleographic. It deals with study of ancient geography during different geological eras and epochs. Maps. Fossils are extensively used as indicators for prehistoric climate, salinity, sunlight and depth of water and availability of oxygen, etc. And as stratigraphic indicators as well. Recently, fossils are used commercially to detect petroleum reserves, coal reserves, gas reserves and reserves of various metal ores. The fossils which indicate the age of the rock in which they are found are called index fossils. The geological timetable. Geologists, as a result of their studies of the strata of sedimentary rocks in the different regions of the world, have classified geologic history into 6 eras. The oldest era with fossils is the Archeozoic era of primitive life, and this is followed in turn by the Proterozoic era of early life, the Paleozoic era of ancient life, the Mesozoic era of medieval life, and the Cenozoic era of modern life. The Paleozoic, Mesozoic, and Cenozoic eras are divided into periods and the periods of the Cenozoic into epochs. There is evidence that between the different eras there were widespread geologic disturbances, called revolutions which raised or lowered vast regions of the Earth's surface and created or eliminated shallow island seas. These revolutions produced great changes in the distribution of sea and land organisms and wiped out many of the previous forms of life. For instance, the Paleozoic era ended with the revolution that raised the Appalachian Mountain and it is believed, killed all but 3% of the forms of life existing then. The Rocky Mountain Revolution, which raised the Andes, Alps and Himalaya as well as the Rockies, abolished most reptiles of the Mesozoic. The following table of geologic timetable shows the eras and some of their subdivisions, the approximate duration of each era, some of the important geological features, and the characteristic animals and plants. Geological timetable. Millions of years ago. Geological feature plants invertebrates vertebrates. Eras periods epochs. Cenozoic Quaternary, recent end of last ice age, decline of woody plants, age of man 70, 0.0. Eras periods. Ordovician Great. Mostly igneous are. Conclusions drawn from fossil record. The most important features of the fossil record which is tabulated in Table 3-1 can be summarized as follows. There existed a multitude of diversified animal groups, similar to the animal groups that exist today. All fossils did not appear at a time, but appeared during different great spans of times. 1. Paleozoic. This piece of rock is from the Silurian period in the Paleozoic era. This era ended 248 million years ago, when more than 90% of species became extinct. The latest theory is that the amount of oxygen in the air fell sharply, suffocating most large, active animals. 2. Mesozoic. Triassic rock, containing ammonites, belongs to the Mesozoic era, the age of dinosaurs. Both dinosaurs and ammonites became extinct at the end of this era, 65 million years ago, when some 25% of species died out. Fossil. Ammonite 3. Cenozoic. This rock, bearing fossil fish, comes from the Tertiary period, part of the Cenozoic era. During the Cenozoic, which is still in progress, mammals and birds have taken over the many vacant slots left by the disappearance of the dinosaurs. After mass extinctions, some surviving species evolve into new forms that repopulate the Earth. Fossil fish. Eat the most primitive forms of underscore. Odd life are found in the oldest rocks. Wing feathers reef G ka. Or remages e ingers IV, moving up through the various auricoid strata, from older to more recent forma R. XK humerus tions, there is a succession of higher and more complex Aldous. Origin of Figure 3.2. Fossil of Archaeopteryx. 1. Transitional forms. The critics of paleontological evidence for the idea of evolution sometimes maintain that the fossil record lacks transitional forms between one major group and another. In fact, fossil record does contain some fossil transitional link forms. One such link is Archaeopteryx, transitional fossil between birds and reptiles. Archaeopteryx was found in the rocks of the Jurassic period and it was about the size of a crow and in certain respects was quite like a reptile. Like birds, it had wings with feathers, but they were small in size. Its tail, quite unlike that of modern birds, was long with a row of feathers on each side. Further, its jaws were equipped with conical teeth and 3 of the digits of each forelimb persisted, armed with claws. It is probable that the forelimbs were used for climbing as well as for flying. However, skeleton framework of Archaeopteryx resembled more with the reptiles than the birds. Thus, Archaeopteryx provides a connecting link between the reptiles and birds, suggesting that birds were evolved from animals like reptiles. 2. Forms showing serial evolution. Fossil records, sometimes, allow us to construct the phylogeny of an animal. Such phylogeny or serial evolution of an animal such as horse, elephant, camel, etc., provides a strong evidence in favor of idea of biological evolution. Phylogeny of horse. The history of horse dates back to about 60 million years in the Eocene period, involving about 20 genera. The horse phylogeny starts with Eohippus hyracotherium, a member of Equidae. The Eohippus lived in the Eocene epoch. It was about the size of a fox terrier dog, but with a longer head. Its legs were short, with 4 toes on each front foot and 3 on each hind foot. The third digit was somewhat longer than the others. All of the toes were placed on the ground and used in walking. Eohippus was a forest dweller and browser, feeding on soft vegetation. The lines of descent perhaps passed from Eohippus through Mesohippus, Myohippus, Parahippus, Merychippus, and Pleohippus to Equus, which represents the modern horse and its allies. Modern horse is plains dweller and a grazer. The most important changes in the evolution of the modern horse from Eohippus are the following. 1. The enlargement and elongation of the third digit, with a loss of the other digits. 2. The elongation of distal parts of the limbs so that the wrist and the ankle are high up. 3. The elongation of the forepart of the skull. 4. The increase of length and mobility of neck. 5. The development of the premolars and molars into high-crowned, continuously growing grinders. And 6. A general increase in body size. The phylogeny of horses is not a freak of nature, as opponents of evolution idea think, but a constantly progressive adaptation history to changing geological changes which compelled horses to undergo modification. Thus, phylogeny of horse and other animals provides evidence in favor of biological evolution. Further, paleontological evidence provides the most direct indication of evolution by showing that through the years organismal types became more complex. It only implies that the simpler forms gave rise to more complex ones. Imperfection of fossil record. It is true that fossils provide direct and strong evidence of occurrence of organic evolution, yet, such a valuable evidence is generally marred by the incompleteness and discontinuity of the fossil record. Thus, about 1.5 million species of living organisms are known today, yet only 130,000 fossil species have been described. Many major groups are not represented as fossils at all. For this, Darwin said that fossil records are like a book whose many pages are missing or torn and whose all letters are not legible. The following factors have been attributed for this discontinuity or imperfection of the fossil record. 1. Problems in excavation of fossils. Since URO Equus L Re Q NH N OA equals OKQ 2 equals Ks 2 I 3 grams I EIJ 7 in. Further, since sedimentary rocks, in which fossils are formed, are mainly developed undersea and major portion of Earth is occupied by sea, therefore, it is not possible to dig out these rocks under the sea. Moreover, it is also not possible to successfully excavate the high mountains, ice-covered areas and very thick jungles, where there are great chances of finding the fossils. Because of these drawbacks, a large number of fossils could not be excavated. 2. Problems of fossilization. Another important factor responsible for discontinuity of fossil record is that of difficulties in the process of fossilization, since majority of animals do not get proper environment for the fossilization due to either of the following reasons. Animals are completely destroyed by predators, oxidizing agents or other factors. Eat the hard parts or either not present in most animals, e.g. invertebrates, or get destroyed by factors such as acidity, etc. Due to this reason, a large number of invertebrates such as protozoans, worms, helminths and arthropods could not be fossilized. IIYE, animals living on land normally. Figure 3.4. Fossil of trilobite get minimum chances of being fossilized, because majority of them do not get any chance to find their way to the sea. IV. The occurrences of entirely unfavorable conditions for the fossilization during various geological periods have contributed to the complete absence of fossils from certain geological periods. V. Population size also affects the fossil record. Thus, species with large populations are more likely to be preserved in the fossil record than population with low densities. V. Even different amounts of calcium deposited in the skeleton modify the likelihood of preservation. Some experts suggest that the sudden appearance of trilobites in the fossil deposits is not because the creatures suddenly evolved but simply because they started using calcium to their shells. A trilobite, Silurian period. 3. Natural destruction of fossils. Lastly, fossils of a variety of organisms are being destroyed due to the following natural causes. I. Enormous amount of heat and pressure results in alteration or complete destruction of the fossils present in deeper rocks. E. Natural upheavals result in the destruction of fossils. E. Changes in course of rivers, landslides, water erosion and floods result in the destruction of rocks and earth crusts containing fossils. IV. Flattening of fossils occurs due to pressure as more and more layers of sediment are piled on top of the bed containing the fossil. RUAYRYO08. Indirect evidences of evolution. Organic evolution. In this chapter, we will discuss the following indirect evidences. Evidences from CIASSIFICation taxonomy. Taxonomy is the science of classification of organisms. It began long before the doctrine of evolution was accepted. Indeed, the founders of scientific taxonomy, Ray and Linnaeus, were firm believers in the fixity, the unchangingness, of species. Linnaeus' classification was based on the principle of like with like. Since the development of the theory of descent with modifications by Lamarck 1809 and Darwin 1859, the organisms were classified on the basis of their interrelationships. Present-day taxonomists are concerned with the naming and describing of species primarily as a means of discovering or clarifying evolutionary relationships. This is based upon the assumption that the degree of resemblance in homologous structures is a measure of the degree of relationship. The characteristics of living things are such that they can be fitted into a hierarchical scheme of categories, each more inclusive. In the last chapter, we have studied direct evidences of D. Analogous structures. Wings of A. Insects, B. Birds, C. Seals and D. Penguins. Then the previous one, species, genera, families, orders, classes and phyla. Thus, very closely related animals or plants are kept in the same species, which shows that they descended from a common ancestor. The species which are much alike are included in one genus which is distinct from other genera. In the same manner, all the like genera are included in one family, which shows again a sign of affinity. Likewise, similar families form orders, and orders are grouped into classes, in which diverse members share only n, all. Very fundamental Chiracteris species is a population of closely similar individuals which form the ticks. Lastly, all the related basic unit of taxonomy classes are included in a phylum. This system of classification of organisms can be best interpreted as proof of evolutionary relationship. If the kinds of animals and plants were not related by evolutionary descent, their characteristics would be present in a confused, random pattern and no such hierarchy of forms could be established. Further, the scheme of classification is itself a proof of descent from a common ancestor as shown by the phylogenetic tree. The forms which are associated together in species, genera, classes or phyla are supposed to have descended from a single common ancestor. For example, the vertebrates like fish, frog, lizard, pigeon, rabbit and man look very different from each other, but they all resemble in having dorsal hollow central nervous system, a notochord and gill slits. This similarity in the outwardly different vertebrate animals indicates their common ancestry. The basic unit of taxonomy is the species, a population of closely similar individuals, which are alike in their morphologic, embryologic, and physiologic characters, which in nature breed only with each other and which have a common ancestry. It is difficult to give a definition of species that is universally applicable. The definition must be modified slightly to include species whose life cycles includes two or more quite different morphological forms, many coelenterates, parasitic worms, larval and adult insects and amphibians. A population that is spread over a wide territory may show local or regional differences which may be called subspecies. Many examples are known in which a species is broken up into a chain of subspecies, each of which differs slightly from its neighbors but interbreeds with them. The subspecies at the two ends of the chain, however, may be so different that they cannot interbreed. Such a series of geographically distributed subspecies is called a Rassenkreis, German, Racecircle. Lastly, the classification of living organisms into well-defined groups is possible because most of the intermediate forms have become extinct. If representatives of every type of animal and plant that have ever lived were still living today, there would be many series of intergrading forms and the division of these into neat taxonomic categories would be difficult. Certain taxonomists have compared the present-day species to the terminal twigs of a tree whose main branches and trunk have disappeared. The main problems of modern taxonomists are to reconstruct the missing branches and to put each twig on the proper branch. Evidences from comparative anatomy. A comparative anatomical study of any particular organ system and the diverse members of a given phylum reveals a basic similarity of form which is varied to some extent from one class to another. The skeletal, muscular, circulatory, and excretory systems of the vertebrates provide especially clear illustrations of this principle, but this is generally true of all systems in all phyla. Such studies in comparative anatomy provide many evidences of biological evolution by showing that anatomical similarities become more and more complex progressively as one proceeds from a lower animal to a higher animal, and all the diverse animals inherited the anatomical similarity from a common ancestor. Examples of anatomical similarities could be multiplied largely, but we restrict ourselves to the few following examples. 1. Connecting link. Animals standing between two groups of animals are called connecting links, i.e., Archaeopteryx and Archaeornis. These extinct animals exhibit both reptilian and avian characters, suggesting evolutionary origin of birds from the reptiles. Among the existing mammals, Monotremata duck-billed platypus and the spiny anteater occupy an intermediate duck-billed platypus as a connecting link between reptiles. Scission between reptiles and typical and mammals. Mammals. For like the mammals they possess hair and suckle their youngs. Reptilian features of monotremes are, they lay large heavily yolked eggs, have a cloaca into which urinogenital ducts and alimentary canal discharge. The shoulder girdle is more like reptiles' presence of interclavicle which is not found in other mammals. Thus, connecting links indicate the path along which the more highly organized groups have progressed during their evolution from more lowly organized ancestral groups. 2. Homology. Homology is the similarity between various organs of different organisms and it is based on common embryonic origin or common ancestry. Homologous organs are those which have the common origin and are built on the same fundamental pattern. But they perform different functions and have different appearance. These organs are variously modified in adaptation to different functions. The theory of evolution suggests that hereditary characters become gradually modified and these modifications make the organism better suited for the changed conditions of life. Homology is found in every organ system from Pisces to mammals. For example, a whale's or seal's front flipper, a bat's wing, a cat's paw, a horse's front leg, a human hand and even a bird's wing, though superficially dissimilar and adapted for quite different functions, viz. Grasping, running, flying, swimming, etc., nevertheless, are homologous structures. Each consists of almost the same number of bones, muscles, nerves, and blood vessels arranged in the same pattern, and their mode of development is very similar. The existence of such homologous organs implies a common evolutionary origin of birds and mammals from some ancient reptilian ancestor. All insect legs are composed of 5 parts that always occur in the same order. Starting at their attachment on the thorax, these parts are coxa, trochanter, femur, tibia, and tarsus. These parts are single segments except the tarsus, which varies from 1 to 5 segments. There have been many adaptive changes in insect legs during the humerus radius carpals. Evolution of the group. Humeru. R equals et fl, phi, 8f, asterisk, legs, such as the first _ie, 5. L, I1 li 1 foot. Il. Airs, the legs off a diving beetle are constructed for swimming, and the claws, pulvili, and hairs of the tarsi of the common housefly make it possible for the fly to walk upside down. The brain of vertebrates presents another good example of homology. The brains of vertebrates ranging from fishes to mammals are constructed of similar parts: olfactory lobes, cerebral hemispheres, optic lobes, cerebellum, medulla and so on. As one moves higher through the series, some lobes become more prominent than others. This happens due to adaptations and evolutionary change. Thus, the cerebral hemispheres are much smaller than the optic lobes in fishes. In mammals they become more prominent, hiding the traces of optic lobes beneath them. This is due to the difference of intelligence between the fish and mammals that the cerebral hemispheres become more developed in the latter. Categories of homology. These are 3 categories of homology. Phylogenetic homology is that existing between different species, e.g., pentadactyl limbs of air-breathing vertebrates. Sexual homology exists in between sexes of the same species, e.g., testes of man and ovaries of a woman, penis of man and clitoris of woman. Serial homology is that existing between organs of the same individuals occupying different levels of the body, e.g., arm and leg of man and appendages of crustacea. In atypical crustacean, there is one pair of appendages borne by each segment of the body and all have a single structural plan, consisting of a basal 2-segmented portion, the protopodite, bearing 2 lateral olfactory outgrowths, the exopodite and incerbrum dopodite. The optic lobe. Opening square bracket, closing square bracket. Cerebellum, medulla oblongata. Structural plan. This similarity is appendages of various body segments: fish, reptile, amphibian, bird, mammal, called serial homology. Perform different functions and accordingly they show modifications in the basic. Fig 4.2. Brains of vertebrates showing homology. Homology in plants. Among plants also, there are many examples of homologous structures. In all seed plants, for example, leaves are homologous structures, as are stems and roots. The many variations in their size, shape, and function indicate the many adaptive modifications that have taken place throughout the group. Leaves modified as thorns, bougainvillea, tendrils, cucurbita, and the parts of flowers, sepals, petals, stamens, and carpels are examples of homologous structures that provide evidence of evolution. In all higher plants, there is a geotropic at-root system and phototropic shoot system. Regarding the latter, natural selection favors b. homologous structures derived from leaves. A. Barrel cactus. B. Heliconia rostrata. the acquisition of a spreading form, rather than a compact form. This is due to positive advantage of trapping the maximum light energy for photosynthetic use. Likewise, the similarities that exist in many __ scapula. Humerus U, A. J. I ulna, F. 3, I. I fused F radius, N 5 inches radius LJ carpus equals I N ulna. N, metacarpus. G. GHPYE hash splint bone. IV. I versus GI canon T equals 1 bone 1 LVVV. Phalanges equals I solid hoof. Fingers 11-15 equals formed by IV split hoof Y1 enlarged. Man dog pig sheep horse fingernail. Figure 4.3. Forelimbs of certain mammals showing serial homology. Analogy, homoplasy. Groups of families of plants in regard to floral parts and symmetry are related to common descent through organic evolution. Thus, in monocots the floral parts are arranged in odd number of 3 or its multiples, e.g., sepals, petals, stamens and lobed pistil. Indicots, the parts of the flowers are commonly in the odd number of 5, very rarely in 3, sandless commonly in the even number of 2 and 4, or their multiples. In the study of comparative anatomy, structures are often found that have the same function and are superficially alike, such as the wing of a bird and wing of a butterfly, yet are quite different in origin and in structural design. Such structures which are non-homologous but with similar functions are said to be analogous organs or structures. They have arisen in the evolutionary process through adaptations of quite different organisms to similar modes of life. The analogy, thus, refers to the relationship between structures which though differ anatomically but have superficial similarity due to similar functions. Often analogous organs have little gross structural resemblance to each other, for example, the gills of Polemon and the lungs of man. Other analogous organs, such as the wings of insects, abs birds, and flying mammals bats, may have a superficial resemblance. Their common function, flight, hastenly one structural solution the evolution of some sort of an airfoil. Analogous organs with a structural resemblance are said be homoplastic. There are many instances where different organisms that are not very closely related have developed similar adaptations for life in similar habitats. This phenomenon is often referred to as convergent evolution, the development of same fusiform type of body for life in the water by Polemon arthropod. N. Radius _ I. Ulna. Wing. A insect B pterodactyl. Shark, fish, ichthyosaurus, reptile, and a dolphin, mammal, illustrates the convergent evolution well. The development of a limbless, cylindrical snake-like body by Ophiosaurus, lizard, Oreotyphlops, Sicilian amphibian, and Typhlops blind snake, for life in the burrows, is another interesting example of convergent evolution in animals. Further, the development of a cactus-like body by members of several different families of plants is another good example of convergent evolution. Among plants, a stem may be modified to appear like leaves and may perform the functions of leaves as in Ruscus. This is a case of analogy. Vestigial organs. Another source of evidence stemming from the studies of comparative anatomy is the presence of organs in various animals that have no apparent function and are useless and often small or lacking some essential part. However, in related organisms, these organs remain full-sized, complete and functional. Such organs are called vestigial or rudimentary organs. The vestigial organs are believed to be remnants of organs which were complete and functional in their ancestors. There are more than 100 vestigial organs in human body. Some common examples of them are the following. 1. Muscles of the external ears. They are nictitating. Membrane equals muscles to FL equals move ears. E. O. Hair on body CBN and LEA _ equals mamma on. Pipe. Third molar. Segmental muscles. Vermiform, on abdomen. Appendix I. Pyramidalis _I, _IEKL muscle vertebrae. Figure 4.5. Some vestigial organs of man. Well developed and functional in quadrupeds, but in man they are useless structures and cannot move the external ears pinna. 2. Panniculus carnosus. There is a well-developed subcutaneous skeletal muscle layer by which some quadrupeds can move their skin at the time of need, i.e., to fly off the irritating insects. This muscle layer is of use in humans. However, it is functional in the forehead by which it can move the eyebrows. 3. Coccyx. Coccyx is the much reduced string of caudal vertebrae and is present at the end of the vertebral column. It is a remnant of the tail present in early embryo of man, but it is shed off much before the adulthood is attained. 4. Vermiform appendix and the cecum. Cecum is located at the point where the large intestine is joined by the small intestine. The cecum is a short pouch, ending blindly except for the small opening into its extension called the vermiform appendix, figure 4.6. The appendix of man is thought to be a remnant of the large cecum, which is cecum large intestine or iniestine. The storage organ for cellosmal loose digestion in herbivointestine rus mammals. In most primates except man, it is of inulcers %%. STTT large size. In human beings it has no function and is ru. Dimentary. It indicates that Tr. Cecum B vermiform appendix C ancestors of man early pre. Figure 4.6. Cecum and vermiform appendix. Rabbit, B man mates, had a much coarse embryo, C, man. Diet due to which cecum was functional. 5. Presence of vestigial mammary glands in male human beings. This might suggest that one-time males also suckle the young. 6. Presence of vestigial O_nictitating membranes. In Manlazert LQTGRT, NN%SR. L. Yanar, the inner angle of each of Rn equals Is is present a little fold of 1,5 flesh called semilunar fold. Plica semilinaris. This core equals. Responds to a structure which. In many lower animals Ismaval horseman. Able third eyelid, called nyctifig. 4.7. Eyes of owl horse and man showing nictitating membrane or. Tating membrane, lying unal sacal. The outer eyelids and. Sweeping across the eye from the inner angle outwards. 7. Presence of wisdom teeth, third molars. These are posterior moss teeth which are variable than the other teeth as regards the size and time of eruption. These are vestigial structures, since they are last to erupt or even fail to erupt. In other primates, third molars are as sound and as fully developed as the rest of the dentition. All these vestigial organs have provided evidences that man has descended from simple primates. Certain other animals also possess the vestigial organs which are as follows. Tiny rudiments of the hind limbs in python and vestiges of pelvic girdle, ilium, and leg bones, femur, in the whale. 2. Wings of Apteryx kiwi, of New Zealand, flightless birds. 3. Reduced third metacarpal, splint bone, in the feet of horse. I. E. Ilium. G. L. 1.2. O. L. Dash dot. I underscore L. Femur I claw. L. GLNTL backbone. 2. Figure 4.8. Vestigial hind limbs of python, snake. All the aforesaid examples provide conclusive evidence in support of the theory of organized evolution as follows. 1. Organisms having homologous and vestigial organs have developed from a common ancestor. 2.0 Organisms with analogous organs had different ancestors. 3. The present-day similarity and dissimilarity of organs is a sort of adaptive radiation to cope with new needs of the environment. Darwin pointed out that both frigate birds and upland geese have webbed feet, yet neither goes into water. He explained their feet as a leftover from their past, both being descended from waterbirds. Evidences from Comparative Embryoiology.